And welcome. Today we look at these two skills for IB biology. Use a table of the genetic code to deduce which codons correspond to which amino acid, and then to use a table of messenger RNA codons and their corresponding amino acids to deduce the sequence of amino acids coded for by a short mRNA strand of known base sequence. So these two are very much interconnected, and let's go in and take a look at what a question actually looks like with the table and how to arrive at the answer. So here you see a typical table from a past paper question here that's provided for this, and this here is an mRNA strand that then has to be read using this table here. Now, one of the things that could confuse students is that although this is mRNA, you don't have to figure out what's the corresponding anticodon and then go to this table. This table is made so that if you take the triplet AUG and you go in here, then you will find the amino acid that is coded for by this. Don't get confused by the fact that in translation an anticodon comes here from transfer RNA, but you're not searching for the anticodon that drags the amino acid into position. You are searching for AUG in this table here. And how do you use this table? Some people look at it and intuitively they figure out how to use it, but I would explain it to you here now. It's A, first letter, so you go here and you identify this box. Second letter U, so you go to the top and you get U, second letter, and you bring down an arrow to see where it intersects with A, and all of this space here is the AU space. Which specific one we need to identify would only be pinpointed when we look at the G and we go to the third letter, which is G, and then we go across this way. And that would say that this first triplet, this first codon, is the start codon. It could also code for the amino acid methionine, but seeing that this is the start of an mRNA strand, let's go with start. Now what you have to do is to keep going and get the rest of the amino acids that are coded for, one triplet at a time. Bear in mind though that there's no overlapping in this code, right? It's AUG and then there's AGC. It's non-overlapping. It's a degenerate code, this genetic code, so there's no overlapping happening. And then also bear in mind that if perchance you arrive at a stop codon at any point in time, the chain is going to stop growing, even if you have other codons in that chain. That stop codon means the chain is going to stop growing. So with that in mind, I would like you to now share the answer for this question, which is to determine the sequence of amino acids that could be translated from this messenger RNA sequence. And this is the messenger RNA sequence, and this is the table to use. Now the answer for this question is start, SER, ARG, ARG, and stop. That is the answer we're searching for. Any of these other answers here could be accepted, but it's definitely not going to be accepted if you put an LEU after that stop codon.